Today I want to talk to you about one of the most groundbreaking medical advancements of the 21st century, about new research that aims to eradicate genetic disease. Genetic diseases occur due to mutated or defective genes. Genes are segments of DNA that direct the cell to produce proteins that perform a wide variety of functions in the body. Many diseases have a genetic basis, meaning the genes provide incorrect instructions to encode proteins, causing the cell to produce abnormal proteins or disable the production of the protein entirely. If a mutated gene causes a necessary protein to be faulty or missing, gene therapy may be able to introduce a functional copy of the gene into the patient's own cells to restore function of the protein and correct the underlying cause of the disease. A gene that is directly inserted into a cell usually does not function. Instead, gene therapy uses a carrier called a vector, a genetically engineered virus that delivers the functional gene into the patient's cells. Retroviral and adenoviral vectors are both currently being evaluated in phase 1 clinical trials, along with adeno-associated virus, AAV, which infects many dividing and non-dividing cell types but has limited DNA capacity. Retroviral vectors can permanently integrate their genetic material into the chromosome of the cell, but require mitotic cell division for transduction. Adenoviral vectors, the common cold, can effectively deliver genes into the nucleus of a wide variety of both dividing and non-dividing cell types, but immunoelimination of infected cells often limits gene expression in vivo. The vector can be injected intravenously directly into the specific tissue in the body, where it is taken up by individual cells. Alternatively, a sample of the patient's hematopoietic or blood stem cells can be removed. The virus is then mixed with the blood stem cells outside of the body. This technique using adenoviral vectors is being used in an attempt to treat both sickle cell disease and severe combined immunodeficiency, or SCID. Sickle cell disease is an inherited blood disorder that affects hemoglobin, the molecule in red blood cells that delivers oxygen to cells throughout the body. The abnormal hemoglobin in people with sickle cell causes the red blood cells to become rigid, causing them to get stuck in the body's small blood vessels, and as a result, the organs cannot get the blood they need. SCID is a rare and life-threatening disease that causes the child to have very little to no immune system, making them severely immunocompromised and thus highly susceptible to infections, as the gene mutation reduces the ability of the white cells in the blood to fight infection. Currently, both of these diseases can be treated with allogeneic bone marrow transplants, but this results in severe side effects, on top of the initial difficulty of finding a compatible donor. This is where gene therapy comes in, using autologous bone marrow transplants to essentially turn the patient into their own donor. This is done by removing the patient's bone marrow, which contains hematopoietic or blood stem cells, using gene editing to remove the mutation, then mixing the adenovirus containing the functional guide RNA strand, enclosed within a protein, with the blood stem cells. The guide RNA recognizes the target DNA strand using the simple rules of ATGC, then introducing the functional DNA strand into the gene, thereby correcting the mutation. The blood stem cells are then reimplanted back into the patient to cure the disease. So gene therapy has the potential to change our relationship with genetic disease. Rather than a patient being born with a potentially life-threatening condition and just constantly treating the symptoms, we now have the potential to permanently cure that patient in so many diseases that we previously never had a cure for. Diseases like hemophilia, blindness, leukemia, and myotubular myopathy, which causes severe muscle weakness, and consequently, children cannot walk or even sit up on their own. All of these have had some success in clinical trials, though gene therapy also holds promise for treating cancer, cystic fibrosis, HIV, diabetes, and heart disease. So this is all fantastic, right? So what are the problems? Firstly, is it safe? The technologies are still relatively new, and with this comes unpredictable risks. Several studies have already shown that gene therapy does pose some serious health risks, such as toxicity, inflammation, and cancer. However, the majority of the side effects do have a simple fix. Medical researchers and regulatory agencies are, of course, working hard to ensure that gene therapy is as safe as possible. Secondly, what are the ethical considerations? How do we decide who gets the treatment? At the moment, research is only targeting life-threatening conditions with no other potential cure. But upon perfecting these treatments, what is to stop us from altering other genes, enhancing them? The genes that determine height, eye color, intelligence, or muscular density. Genetic enhancement has always been a controversial issue, but here we are looking to the future. where, for example, traveling to space and colonizing Mars is becoming increasingly likely for human beings. However, studies have shown the impact that extended periods of time in space can have on the human body, such as bone and muscle loss. So, could it be that we need to make these kind of enhancements in order to allow people to live there? And of course, going to space isn't the only consideration. So, while ethically, curing genetic diseases is a no-brainer, technology is always advancing and the future is fast approaching. And we as a society need to decide what we want to do with it and where we want to go. Thank you.